For decades, scientists thought they understood what happens to the brain when the heart stops. They were wrong. The human brain does not shut down at the moment of death. It surges. Gamma oscillations, the fastest brain waves the brain produces, the ones most closely linked to conscious experience, spike to levels 5 to 8 times stronger than anything measured during normal waking life. This was recorded in dying humans. In 2023, the brain's final act is not silence. It is the most intense electrical activity it will ever produce. And the physics of why this happens, what is actually occurring inside 86 billion neurons as oxygen runs out, may be the strangest thing in all of neuroscience. Death is supposed to follow a clean sequence. Heart stops. Blood pressure drops to zero. The brain, cut off from its oxygen supply, loses electrical activity within seconds. Measurable brain function ceases. Death is declared. That is the textbook version. It is orderly. It is final. The brain does not follow it. A neurophysiologist named Jimo Borjigin had been studying the boundary between life and death for years at the University of Michigan. Her primary research was in something entirely different: how the brain regulates hormones and circadian rhythms, the chemistry of waking and sleeping, the molecular machinery that makes you alert at noon and drowsy at midnight. But something kept appearing in her data that she could not explain. In animals that died during experiments, animals being studied for other purposes entirely, her instruments occasionally caught unusual electrical signatures at the moment of death. Spikes of organized activity in brains that should have been shutting down. Most researchers would have noted the anomaly and moved on. It was not what they were looking for. Borjigin did not move on. In 2013, she designed an experiment specifically to look at what happens in the brain at the moment of death. She attached high-density EEG arrays to 9 rats and monitored their brain activity continuously, before, during, and after cardiac arrest. Every electrode recorded, every frequency band tracked. She wanted to see what the dying brain actually does, with no assumptions about what it should do. What appeared in the 30 seconds after cardiac arrest was not the expected decline into silence. It was a surge, gamma oscillations. Brain waves cycling between 30 and 100 Hz spiked sharply within half a minute of the heart stopping. Not scattered random noise. Coherent, synchronized activity across multiple brain regions in patterns that neuroscience associates with one specific thing: conscious processing. The power of these oscillations was 5 to 8 times stronger than the waking baseline. The dying brain was not dimming. It was more electrically active than it had ever been while alive. 5 to 8 times. A brain with no oxygen supply, no heartbeat, no blood flow, producing organized electrical activity at levels that dwarf normal consciousness. Borjigin published the findings in the Proceedings of the National Academy of Sciences. The response from the neuroscience community was immediate and divided. The data was meticulous. 9 rats, all showing the same pattern: a sharp, coherent gamma surge within 30 seconds of cardiac arrest, concentrated in cortical regions at power levels that dwarfed the waking state. No one disputed the measurements. What they disputed was the interpretation. Rats are not humans. A rodent brain dying under controlled laboratory conditions, anesthetized, monitored, with cardiac arrest induced by injection, may not reflect what happens in a human brain dying of heart failure or stroke or organ failure or old age. The gamma surge was real. Whether it meant anything for human death was unknown. Other researchers pointed to a simpler explanation. Perhaps the surge was just a random firing of neurons losing their energy supply, an electrical spasm, not organized processing, the dying gasp of a machine breaking down. Borjigin's data showed coherence and frequency patterns that argued against randomness. But the skeptics had a point. Coherence in a rat cortex does not prove consciousness in a rat cortex, let alone a human one. The finding was provocative, suggestive, and fundamentally limited by the fact that you cannot ask a rat what it experienced. So she spent the next 10 years trying to answer the only question that mattered. Does this happen in humans too? Getting from rats to humans took a decade. You cannot induce cardiac arrest in a healthy person for research purposes. Borzygin needed dying patients whose families would consent to EEG monitoring during the final moments, a request that requires extraordinary sensitivity and trust. It took years of collaboration with intensive care physicians and palliative care teams. Eventually, 4 patients at the University of Michigan Hospital, all on life support with no prospect of recovery, all with families who had made the decision to withdraw care, agreed to participate. With consent and ethical approval, Borzygin's team placed the same type of high-density EEG electrodes on the patients' skulls that she had used on the rats a decade earlier. The electrodes recorded continuously as the ventilators were turned off, as the heart slowed, struggled, and stopped. 2 of the 4 patients showed it. After cardiac arrest, after the heart had stopped and blood was no longer reaching the brain, organized gamma oscillations appeared, not scattered randomly across the cortex, which would suggest disorganized electrical noise from a system breaking down. Concentrated in a specific anatomical region, the temporoparieto-occipital junction, where the temporal, parietal, and occipital lobes converge at the back of the brain. You see, this location matters. Giulio Tononi at the University of Wisconsin and Christoph Koch at the Allen Institute for Brain Science had spent years mapping the neural correlates of consciousness, identifying which brain regions are active when a person is consciously aware versus when they are not. Through hundreds of studies using fMRI, EEG, and direct cortical stimulation, they converged on this posterior region as the area most consistently linked to conscious experience. They called it the posterior cortical hot zone. It is where the brain integrates visual information, spatial awareness, bodily sensation, and memory into a unified experience. The part of the neural architecture that generates the sense of being someone, somewhere, experiencing something. Damage to this region disrupts conscious experience even when the rest of the brain is intact. Stimulation of this region produces vivid perceptual experiences even in patients under anesthesia, the part of the brain that makes you feel like you. In the dying patients, it was firing at levels that exceeded normal waking consciousness. For between 2 and 5 minutes after the heart stopped, published in 2023, a decade after the rat study, the same researcher, the same question, now answered in human beings. 2 of 4 patients. A small sample. Borzygin is the first to acknowledge this, but the 2 who showed the surge showed it in the exact brain region, at the exact frequency band, with the exact coherence patterns that the neuroscience of consciousness would predict if those dying brains were generating something resembling experience. Not proof of consciousness after death, but not the quiet flatline that medicine assumed. Now, to understand why the dying brain does this, why it erupts instead of fading, you need to understand the single most expensive operation the brain performs. Every neuron in your brain maintains a voltage difference across its membrane, a charge separation between the inside of the cell and the outside. The inside sits at roughly minus 70 millivolts relative to the outside. That is a small number. A single AAA battery produces about 2,000 times more voltage, but that tiny gradient is everything. Maintained simultaneously across 86 billion neurons, it is what makes thought possible. Every sensation you have ever felt, every memory you have ever recalled, every face you have ever recognized, every word you are hearing right now, all of it exists as patterns of these voltage gradients changing in coordinated sequences across neural networks. Consciousness, as far as physics can describe it, is a pattern of voltage changes. One protein is responsible for maintaining this gradient, the sodium-potassium ATPase pump, a molecular machine embedded in the membrane of every neuron. It burns ATP, the cell's energy currency, to continuously push 3 sodium ions out of the cell for every 2 potassium ions it pulls in. That is all it does. Push ions in, push ions out, maintain the charge. And it runs without stopping from the moment a neuron forms until the moment it dies. It is astonishingly expensive. It consumes roughly 40% of your brain's total energy budget. Your brain uses 20% of your body's total oxygen supply, despite being only 2% of your body weight. And nearly half of that oxygen goes to running these pumps alone. Nothing else in the body costs this much to maintain. 40% of the energy of the most energy-hungry organ in your body, spent on one task, keeping the voltage at minus 70 millivolts, keeping you conscious. When your heart stops and oxygen delivery ceases, your pumps begin to fail. They do not stop instantly. Your brain stores a small reserve of energy, roughly 20 seconds worth, in dissolved oxygen bound to hemoglobin in the blood vessels threading through your cortex, plus glycogen stored in astrocytes, the support cells that surround and feed your neurons. 20 seconds. That is the entire buffer between a beating heart and the beginning of cellular collapse. During those 20 seconds, the pumps run on fumes, burning through the last ATP molecules, maintaining the voltage gradient that makes your consciousness possible, spending your brain's final reserves on the same task they have performed since you were born. Every neuron in those 20 seconds is still working, still maintaining minus 70 millivolts, still you. Then the ATP runs out. The pumps stop, and the carefully maintained voltage gradients that took 40% of your brain's energy to sustain begin to collapse. Not everywhere simultaneously, but in a wave. What happens next is called spreading depolarization, sometimes called a brain tsunami. As each of your neurons loses its membrane potential, collapsing from minus 70 millivolts towards zero, the ion channels that were held shut by the voltage gradient swing open. Sodium floods into your cells. Potassium floods out. The careful separation that kept each neuron charged and ready to fire dissolves. And this collapse does not happen everywhere at once. It propagates from neuron to neuron in a slow-moving wave, 2 to 5 millimeters per minute, spreading across your cortex like a rolling blackout across a continental power grid. Jens Dreyer and colleagues documented these waves directly in dying human patients in 2018, recording the exact electrical signature of this cascading failure as it moved slowly across the surface of the brain. A slow wave of electrochemical collapse rolling across your 86 billion neurons. Every cell it reaches dumps its contents. Here is what no one expected. The wave is not just destruction. As each neuron's membrane potential collapses, the sudden voltage change triggers a burst of firing, one last action potential or a rapid series of them before the cell goes silent permanently. Multiply that by billions of your neurons depolarizing in sequence, and the wave of death becomes, paradoxically, a wave of activation. Gamma frequency activity. Organized, coherent, concentrated in the posterior hot zone, the part of your brain that generates the experience of being you. Your brain's dying wave is also, briefly, the most intense moment of coordinated electrical activity it has ever produced. And this is where the chemistry shifts into something no one anticipated. As each of your neurons depolarizes, it releases its stored neurotransmitters. Not in the controlled, targeted way that normal signaling works, where a precise quantity of transmitter crosses a precise synapse to activate a precise target, but everything at once. The entire stored supply dumped into the spaces between your cells. Glutamate, your brain's primary excitatory neurotransmitter, floods out in concentrations that would be toxic under normal circumstances. The glutamate activates neighboring neurons that have not yet depolarized, which fire and release their own stores, creating a chemical cascade racing just ahead of the spreading wave. But glutamate is only part of the flood. Serotonin, the neurotransmitter that shapes your mood, your perception, your sense of what is real, is released in quantities that far exceed anything that occurs during normal brain function. And serotonin, at these concentrations, saturates a specific receptor on your cortical neurons, the 5-HT2A receptor. This receptor is significant because it is the gateway through which the brain's most profound alterations of conscious experience are produced. When this receptor is activated at high levels, the result, in living brains, is a radical shift in perception dissolution of the boundary between self and world, a sense that what is being experienced is more real than ordinary waking life, encounters with presences, panoramic memory. The dying brain does not need any compound from outside. It floods this receptor with its own serotonin, produced by its own collapse. Whether this produces anything resembling conscious experience in a dying person, nobody can say with certainty. Some researchers have proposed that the brain may release its own consciousness-altering compounds at the moment of death, endogenous molecules that activate the same receptor system the serotonin flood is already saturating. Dean and colleagues confirmed in 2019 that one such compound, DMT, is naturally present in living rat brain tissue. Whether it surges at death in humans has never been measured, but the broader chemistry does not depend on any single molecule. The serotonin flood alone, measured, documented, pharmacologically inevitable at the concentrations produced by mass depolarization, is sufficient to saturate the 5-HT2A receptors across your cortex. Your dying brain is drenched in its own consciousness-altering chemistry. Same receptor, same neurotransmitter system. Activated not by a molecule from outside, but by the brain's own collapse. Which raises a question that the data cannot fully answer but cannot be ignored. Do dying people experience something? Pim van Lommel, a Dutch cardiologist, spent years resuscitating cardiac arrest patients. Some of them came back with memories, not confused fragments, vivid, detailed, structured experiences from the period when their hearts had stopped and their brains should have been producing nothing at all. Van Lommel decided to study this systematically. He enrolled 344 cardiac arrest survivors in a prospective study, published in The Lancet in 2001, and interviewed each one within days of resuscitation. 62 of them, roughly 18%, reported conscious experiences from the period of clinical death. What they described was remarkably consistent across patients who had never met each other, across countries, across belief systems, a sensation of moving through a dark space toward warmth and light. A life review, not a highlight reel, but their entire lives experienced simultaneously, as if every moment existed at once, seen not just from their own perspective, but from the perspectives of people they had affected. Encounters with deceased family members, a spouse, a parent, a sibling, recognized immediately, described with a specificity and emotional clarity that the patient said exceeded anything in ordinary memory, and a sense of peace. Nearly every patient who reported an experience described it the same way, the most profound peace they had ever felt. Several said they had not wanted to return, that whatever they had experienced felt more real, not less, than the life they came back to. Van Lommel found no correlation between these experiences and the medications patients had received, the duration of their cardiac arrest, their age, their education, or their religious background. Lifelong atheists reported the same phenomenology as lifelong believers. The experiences did not match what oxygen deprivation alone would predict. Hypoxic hallucinations are typically confused, frightening, and fragmented, not peaceful, structured, and coherent. These are not easy findings to dismiss. They are also not proof of anything beyond the experience itself. Anecdote, a skeptic would say. Memory fabrication. Oxygen-deprived confabulation. Sam Parnia spent the next 2 decades testing exactly that objection. His AWARE studies, the largest scientific investigation of consciousness during cardiac arrest ever conducted, were designed to determine whether patients' reported experiences corresponded to real events. AWARE 2, published in 2023, found that some cardiac arrest survivors recall specific auditory and visual details from their resuscitations, things said by doctors, equipment used, actions taken during CPR. That corresponded to verified events occurring during periods when the EEG showed no cortical activity and the heart had stopped beating. Parnia's conclusion, stated with the caution of someone who has spent 20 years collecting data on a question that most of medicine would prefer not to ask, consciousness during cardiac arrest may be more common than clinical medicine has assumed. The patients who recalled events were not describing dreams reconstructed after waking. They were describing specific, verifiable, real-time events. A nurse's words, the sound of a defibrillator charging, the sequence of chest compressions, from a period when every clinical instrument said they should have been experiencing nothing at all. The gamma surge provides a possible mechanism. The neurotransmitter flood provides a possible trigger. The NDE phenomenology provides a possible description of what the experience feels like from the inside. None of these close the question. All of them open it wider. Borjigин's gamma surge data. The neurotransmitter flood documented in the spreading depolarization literature. The NDE reports from Van Lommel. The verified recall from Pania. None of these individually proves that dying people have conscious experiences. Together, they describe what your brain will do when your heart stops. Not shut down in an orderly way, but produce, for a brief window of 2 to 5 minutes, the most intense, organized, neurochemically saturated activity it has ever generated. Concentrated in the exact region of your cortex associated with conscious experience, flooding the exact receptor system that produces the most profound alterations of consciousness known to pharmacology. This is not what the textbook says will happen to you. The textbook says your brain loses oxygen, the electrical activity fades, you are gone. A clean sequence, an orderly shutdown. What the data shows is something different. A brain that does not go quietly. A brain that produces, in its final minutes, a surge of organized activity in the precise region associated with consciousness, at frequencies that exceed anything it generated during decades of waking life. Whether that activity constitutes experience, whether you will be there, inside that surge, feeling something, witnessing something, is a question that physics and neuroscience can describe but cannot yet answer. The instruments can measure the voltage. They cannot measure what it is like to be inside it. Life and death are not separated by a line. They are separated by a gradient. And somewhere in that gradient, your brain does something that 86 billion neurons spent your entire lifetime preparing for. It releases everything, all at once, in a storm of electrical and chemical activity that we are only beginning to measure. Whether anyone is there to experience that storm is a question neuroscience cannot yet answer. Minus 70 millivolts. That is the voltage at which a human neuron maintains its resting state. The charge that makes thought, sensation, memory, and consciousness possible. 0 millivolts. That is the voltage at which that same neuron completes its depolarization in the spreading wave of death. The distance between those two numbers is where the entire experience of being alive happens. Every thought you have ever had, every face you have ever recognized, every memory you have ever recalled, every moment that felt like it mattered. All of it exists in that 70 millivolt window, maintained by a pump that consumes 40% of the most expensive organ in your body, every second of every day, without stopping, from the moment you are born. When the pump stops, the window closes. It takes between 2 and 5 minutes.